So good morning, everyone. Thanks for the opportunity to talk about hop latent viroid. My name's Zamir Punja, and I'd like to start with just an acknowledgement of our collaborators and co-authors on this particular presentation. So this is greenhouse-grown cannabis, and this is the production system that I'll be talking about with regard to spread of hop latent viroid. It's basically greenhouse-grown cannabis. And hop latent viroid is a major problem, not just in Canada, but as many of you know, also in the US. So I'm going to start out with some symptomology pictures just to indicate what this viroid actually does to cannabis plants. So these are obviously high THC lines. And what you see here on the left is the first symptomology that you would encounter in a vegetative plant that is affected by hop latent viroid. And as you can see, the stunting, the shorter internodes, in some cases, the leaves are slightly darker green than the healthy plant that's over on the right. So this would be in highly susceptible genotypes. You don't necessarily see those symptoms in every cannabis variety or strain that's grown. Here it is a little bit later on when the plants are starting to go into flower. So this would be 2 weeks into flower. And again, you can see the plants are much smaller. The leaves are dark green. And as you'll see in a few minutes, the floral development is significantly reduced. And so growers do recognize symptomology of hop latent early on, particularly in highly susceptible varieties. So this is what obviously is the major economic impact, is when hop latent viroid infected plants going into flower have much shorter inflorescences, poorly developed, as you see here. And as you'll see later on, the yield goes down, and so does the levels of cannabinoids, in particular THC and CBD. So this is, again, a very obvious symptom on highly susceptible genotypes. When the growers walk through, they'll see the stunting, and obviously the term dudding has been used to describe this poor development of the inflorescence. Now, we also looked at trichomes, and as all of you know, trichomes are obviously the most important feature in cannabis plants that allow the production and accumulation of cannabinoids, including THC. And here you see a very nice comparison between a healthy extract of trichomes. So these were trichomes that were collected from dried flowers right before sale. And you can see the impact of hop latent on the production of trichomes. They're smaller, and they're not as well developed as they should be. And in fact, when you look at it under the scanning electron microscope, you will see that healthy compared to infected, the actual trichome numbers aren't different. What you see is a smaller development of the trichome glands, or the heads, and as you'll see in a minute, reduced production of the cannabinoids. So there's a direct impact on trichome development as a result of infection by hop latent. So here it is, a dramatic comparison, the same variety, the same genotype, infected versus healthy. The healthy, large glandular heads full of cannabinoids compared to the shriveled, shrunken heads that are in hop latent viroid infected plants. And these are at the same magnification. So not just a reduction in size, but also reduction in volume. And obviously this has commercial impact to growers, because in Canada anyway, THC levels are driving the economic value of cannabis. And so here's some data to illustrate not just the trichome head size, but also the length of the stalks is reduced. And of course, the most important feature is THC content. And as you see here on the right, THC content goes down from approximately 18% in this genotype to about 11%. So almost a 40% reduction in the production of THC specifically. So again, not really sure what the impact is on the trichomes directly. Could be an indirect effect with poor growth, or they could be a shutdown in some of the metabolic pathways leading up to THC production. Detection of hop latent viroid is fairly straightforward. Most commercial labs have developed PCR primers and kits that will detect this viroid, 256 base pairs, and as you heard in the first talk this morning, you get the dimers, the monomers, the dimers, the trimers, and so on. We tend to see this multiple banding pattern in very susceptible and highly infected plants, which suggests that the replication is going at a pretty good rate, and these dimers and trimers accumulate as a result. You notice in this plant that the viroid is everywhere, going from the lower leaf to the middle leaf to the top leaf, as well as in the roots. So in this case, this is a systemically infected plant. So what I'll do today is I'm going to talk to you about what this viroid does, how it moves, first of all, from mother plant to cuttings, from pruning wounds into stem infections. I'm going to talk about movement from stems into roots, from roots throughout the plant, causing systemic infection. I'm going to talk about spread through water. A question came up earlier this morning. How does this thing spread naturally? It spreads through water. It spreads through flowers, both male and female. It spreads into trichomes. It spreads into seeds. And as you'll see at the end, we've also confirmed that tobacco and tomato plants are susceptible to hop latent. So there's a lot of material here. Most of it is bad news in the sense that this viroid really is a stable entity capable of extremely rapid spread. But we will talk about management methods as I get towards the end. Okay, so one of the major problems, of course, is mother plants, which are used to derive cuttings, can be systemically infected, as you see here, all the way through, but yet not show any symptoms. So that mother plant isn't reduced in size. It doesn't have any physical manifestation that it's infected. There's no stunting. There's no yellowing. And these plants are maintained, obviously, by the growers because that's where they make the cuttings from. But yet, when you look at the tissues in the plant, the viroid is present everywhere, in the roots, in the leaves, in the stems, and so on. We do know, and others have observed too, that when you take cuttings from these infected plants, the cuttings don't root as well. And so here again you see very small root production as opposed to the healthy cutting. And there's some theories about that, certainly gene silencing and other mechanisms that would explain reduced root growth could be occurring in these plants. And so when you start out with a cutting that has fewer roots, then clearly it's not going to be a plant that yields as well. This shows you the influence of cuttings from 3 different genotypes 14 days after rooting was initiated on transmission of the viroid. And lo and behold, every single cutting from every single genotype that started from an infected mother is infected with hop latent viroid. You can see the very, very consistent banding patterns. And so this is obviously a very, very major concern for growers in that your cuttings from an infected mother are all going to be infected if the plant was systemic initially. So within 14 days after the cuttings are initiated, we can take the roots, and every one of them has hop latent. We've also done mechanical transmission studies, and what we do here is try to emulate what growers would do, and that is when they take cuttings with shears, they leave a wound site such as that. And that would be a practical way to demonstrate transmission. And so we did that. We took infectious sap and added it to that exposed cut stem and monitored the spread of the viroid. And what you see here is that at points 1 and 6, so point 1 being the top of the plant and 6 being the roots, are the 2 areas that first picked up the viroid when the inoculation was made roughly here. So a little bit of spread apparently up into this area, but mostly down into the roots, as you see here in number 6. So this is after 2 weeks. And then when you monitor this over time, what you start to see at 4 weeks is that we are starting to see much more spread of the viroid, and not just now into the roots, which is the arrows, but also into the leaves. So during the period of 2 to 4 weeks, it's now spreading from the bottom of the plant up into the rest of the foliage in that particular direction, regardless of the genotype that you use. And then when you get to 6 weeks, every single part of the plant is infected. So once again, very uniform infection here in different genotypes over time. The L is leaf and R is root. So here's the theory then, is if you introduce it here at a point of inoculation, you are going to see a downward spread, which is the yellow arrows. Once it gets down into roots in 2 weeks, you're going to see upward spread, the red arrows. And depending on where you sample, in a systemic plant, 4 weeks, you'll see it up here. And then 6 weeks, it's through the entire plant. This does vary by genotype. It varies by environment. It varies by other factors. But this indicates that in a mother plant, from a cutting, in 6 weeks, that plant's going to be systemically infected. And this is not different from what you would see in the literature with regard to how viruses and viroids spread. From a given point of inoculation, you can see the arrows, some of the arrows going up, but primarily going down into the root zone and then up back into the tissues. And so this reflects transport through the phloem as well as transport through the plasmodesmata from cell to cell. Very consistent with what's known for other viruses and viroids in plants. We don't think that mechanical transmission, and that is lateral spread, is a big deal or important in this viroid cannabis. So this just shows you the systemic spread from inoculation. 2 plants right next to one another, rubbing against another, a person touching this plant, moving over to this plant. We don't see a lot of transmission in that fashion, which is really, really good news because this is not a TMV. This doesn't spread like TMV. It appears that it does need a pretty good wound site on a stem, or as you'll see later on in the root, in order for it to transmit. Mechanical transmission through simply rubbing or plants being next to one another does not seem to transmit this viroid. And that would explain what growers have seen. They don't see these outbreaks as a result of mechanical transmission. Okay, so this is just a summary. Pruning wounds seem to be very important. Physical abrasions of leaves, plants brushing up against one another does not seem to be important for spread. And certainly pruning tools seem to be the most likely way in which this viroid is spreading in a cannabis facility. The other thing we wanted to demonstrate was that root transmission is possible. So what we did here was we took an infected plant and a healthy plant, grew them in these cloners. They were physically separated from one another, except for the hydroponic solution that was floating around these plants. And within 2 weeks, the plant on the right started to pick up the viroid, suggesting that movement through the nutrient solution was possible. We repeated the experiment in the greenhouse environment, and here, infected plants, healthy plant, physically separated. We flooded this tray 3 times a day or twice a day and monitored to see spread. And within 2 weeks, 20% spread occurred from the infected root system over to the other side. 20% is significant when you have 1 million plants. It may not sound like a lot in terms of numbers, but it's pretty significant. So this viroid is probably getting sloughed off in root cells, maybe in the root cap, in the root zone. We don't think that it's necessarily viruses that are exuded out or viroid that's exuded out. It's probably fragments of roots, as you see here, floating around in the nutrient solution that is allowing this viroid to spread. We've also detected hop latent viroid in many places in the growing facility. This area here under the root zone on the table contains hop latent viroid. These emitters on the irrigation system has hop latent viroid, and certainly root fragments that come off the plants have hop latent viroid. Recirculated water, as I'll show you in the next slide, also has presence of the viroid. So this thing moves quite well on the surface of tables between plants that are being shared with the same irrigation system. And here you see recirculated irrigation water, very faint but positive bands showing that the viroid is present in this material. We've also done qPCR with this work, and as I mentioned earlier, the area right underneath the slabs of the plants right here, the reading on a qPCR machine was around 25, which is fairly significant, almost equivalent to what you see in the roots. And then swabs of the table surface, water samples, residual water, and recirculated water are all positive for hop latent viroid. And this is our estimate to try to determine exactly how many viroid particles we're dealing with. This was done with synthetically derived RNA, trying to explain how the qPCR readings correlate with viroid concentrations. And so approximately a qPCR reading of around 20 gives you about 1 million viroid particles. So we're looking at least 100,000 or so particles or fragments of RNA present on those tabletops. Again, going through the literature, it's not surprising. There are many viruses and even viroids that are transmitted through water. Potato spindle tuberviroid, for example, when you grow these plants next to one another, a healthy plant here or infected plant here, a healthy plant there, you will see lateral transmission through roots, through water, through various other systems. And so it's not unique to this particular viroid or to this particular host pathogen system. Other viroids do similar things as far as movement through water. We were surprised with this finding. This is a situation where my student, because they wanted to save on the environment and economics, they used these disposable pestles, which is supposed to be used once and thrown away, but they decided to hang on to them and autoclave them. And after every cycle of autoclaving, we checked to see whether the hop latent viroid was present. And lo and behold, a cycle of autoclaving does not destroy this particular viroid. We also found it in the buckets that we use to store our nutrient solution. We found it on the surface of these handheld watering cans. These are all positive for hop latent viroids. So something to be aware. This thing does stick around on utensils and equipment in the lab. Okay, so what happens when a cannabis plant goes into flower? As most of you probably know, in order to flower a cannabis plant, you have to move it over to a 12-12 cycle. And we were able to show that when you do that, look at the presence of hop latent viroid, FL. FL stands for flower. So much so that when you compare the movement of hop latent viroid in a plant that's flowering compared to a plant that's still in vegetative growth after 2 weeks, you do find it in the roots at 24 hours. But look what happens in the 12-12 cycle. Every single part of the plant is infected. So something has happened in the process of flowering, the physiological changes that occur during a flowering cycle that actually causes this viroid to spread and multiply and replicate considerably into these flowering tissues. And here again is just some data, qPCR data, to show you that when you look at a plant that's under a 12-12 cycle, the qPCR numbers are way, way lower than they are when the plant is under a 24-hour cycle. So again, suggesting the shift over, the physiological shift into flowering is causing this viroid to replicate, to move, and potentially become symptomatic. We looked at dried flower samples that would be making their way to market, and guess what? Hop latent viroid is present. Not surprising. If it's moving up into the flowers, it's also going to be in the dried flower samples. So as I mentioned earlier, these are the trichomes that are produced on flowering tissues of cannabis plants. And so the next question we asked, can this thing move into the trichomes? So we made collections of trichomes from infected plants as well as from healthy plants, and looked at them to make sure they were pure trichome preparations, as you see here. We then extracted the RNA to see whether hop latent viroid was present. And lo and behold, hop latent viroid is present in the trichomes that are extracted from these plants that are infected. Here are the trichome glands, a beautiful image showing you these bulbs that happen. And the arrow basically tells you that as the plant goes into flower, the viroid is actually moving along into these particular tissues, which is perhaps not surprising given how easy it is to spread in a plant, but still quite surprising for us. And again, if you look at the literature, one can find lots of reports of where trichome formation is a photosynthetic sink. There's a lot of energy demand that goes into these trichome development, and it's simply a matter of the viroid finding its way through there as the photosynthates flow from the bract tissues into trichomes. That would explain why we're seeing so much of it there. So much so that even the resin that is extracted from these infected trichomes has hop latent viroid in it. So these are different resin extraction methods, mostly CO2 extraction, and the viroid is present. I don't know what the significance of that is, but it at least tells you how stable this thing is. It can make its way through a CO2 extraction protocol. Okay, so the viroid does move into male flowers. If you induce male flowering in cannabis plants, you will see the presence of the bands, not just in the male flowers, but also in the anthers. So it does move into male flowers. It's also present in the pollen. These are pollen grains. Here's the presence of the viroid in pollen grains, in anthers, as well as the actual pollen itself. And of course, if it's there, it's going to be in the seed. It's also present in both hemp seed as well as cannabis seed. There's a 256 band, a very high frequency of transmission into seeds. And you do see an impact of the viroid on seed development, which again is not surprising. These are seeds from an infected plant, seeds from a healthy plant. They're not going to germinate and grow as well as you would in the healthy. So it is devastating in that regard. We also had some samples of thrips that were feeding on an infected plant, and when we crushed up the thrips and did an RNA extraction, it's present in thrips. So maybe thrips are transmitting hop latent viroid. It's quite possible. We germinated some of these infected seeds, and I wanted to show you that as the seeds start to germinate, the viroid is present on the seed coat, and the first place we find it actually is in the leaves, in the cotyledons, and as the leaves develop, when the leaves are fully formed, it then moves down into the roots. So you don't see it early on here. You see it in there, and then you see it move down from a seedling. So you do see seed transmission as a result of infected seed. So these are some of the experiments we have underway looking at host range. These are the various plants we're looking at. And the way we did the inoculations was simply make a cutting or remove a leaf on a tobacco plant or a tomato plant and add the sap to these injured tissues. And over time, you do see the presence of hop latent viroid in tobacco leaves. This is after 3 weeks, and in tobacco roots after 11 weeks. It moves a lot slower in these plants than it does in cannabis, where you see movement in about 2 weeks. This takes a lot longer. And similarly in tomato, it takes about 8 weeks before we actually see high levels of the viroid in the roots. But certainly, experimentally, the viroid can infect these plants. What does it mean from a practical perspective? I don't know. I'm not going to put the word out there that growers should freak out and worry about alternative hosts. Not yet. These are under experimental inoculation conditions. Okay, so finally, I'm just going to talk quickly about factors that destabilize hop latent viroid. We looked at a number of things, temperatures, UVC, various treatments of various chemicals, as well as addition of various enzymes. I'm just going to go through this fairly quickly here. This is where we tested infected roots with various treatments, Virkon, xeritol, bleach, hypochlorous acid. At these concentrations, the viroid is extremely stable in the roots, and you cannot eradicate it from those tissues. We looked at temperature, once again, going from 30 degrees all the way to 70 centigrade, 15 to 30-minute exposures. The viroid is still stable. The upper bands here, which are probably the linear bands, are removed, but notice, heat stable, extremely heat stable. We looked at dried sap and leaves survival up to 4 weeks, just lying down on the tabletop or in the lab. And here we also looked at UVC treatment. Again, very stable. You can't destroy this viroid with UVC. And of course, if you add enzymes such as RNases, you can remove the viroid totally. This is RNase A, which cleaves as an endonuclease enzyme, and you can certainly eradicate hop latent with that. Okay, so finally, we wanted to develop a methodology that would allow us to test various chemicals for destruction of hop latent viroids. So this just shows you we extracted sap, we collected it in an Eppendorf tube, added particular chemicals that we wanted to test to see if it destroyed the RNA, and then we ran a gel to see what the effect would be. And the results are presented in the following slides. These are various treatments that we looked at, Virkon, bleach, RNase, and various other things. The arrow points specifically to bleach. 10% bleach has a destructive effect on the RNA itself. Here's again a whole series of these treatments. So this was the infectious sap. These were the treatments that were added. And where you see the absence of bands, again, corresponds to bleach, 10% for 2 minutes, 20% bleach for 2 minutes, 20% for 1 minute, and the RNA is destroyed. And then we also looked at hypochlorous acid. So hypochlorous acid does destroy the viroid. Notice that Virkon does not. No effect on the RNA at all. It's mostly the bleach and the hypochlorous acid. Okay, so conclusions. This viroid spreads extremely well through cuttings. It spreads to the plant, becomes systemic. It's present in female flowers. It's present in trichomes. It's present in male flowers. It's present in seeds. It causes a huge reduction in THC levels. It's very stable in dried leaves. It's stable in sap. And the only way we could destroy the RNA was exposure to 10% bleach and 1,000 ppm hypochlorous acid for a minute. So disinfection of tools are going to be relying on these two particular chemicals in particular. And we do know that there are significant genotypic differences in response to hop latent viroids. So that requires a little additional work. Some questions? We don't know what levels of viroid actually are needed to cause infection. Is it 1,000? Is it 10,000? Is it 1,000,000? And how do the viroid levels correlate with disease symptoms or latency? And again, of course, what environmental factors are required. So I'm going to end here once again with the acknowledgement of the sponsors. We're out of time, so I'm going to be available during coffee break or during lunch for any questions that come up. And so I hope I haven't depressed you all with this news, but it is a challenge, but we'll get to it. We'll figure it out. Thank you.